Thank you. Our next talk would be by Alan Norrbom, and new phylogeny of Tephritidae Diptera, data based on nuclear loci obtained by.
Thank you, Severin. When I first wrote this, When I first wrote this title, the plan was for me to talk about just Tephritidae phylogeny, but based on the results, as you'll see, it makes more sense that I'm also covering the Tenostylids as well as the Pyrgodids. As Keith was explaining, he and Brian and I are listed as the authors of the presentation, but this work is really a result of a large team throughout the world, without whom it wouldn't be possible. Although Tephritidae classification has changed tremendously since I started being involved with them, there's still a lot we don't know about Tephritidae phylogeny. Most of the current knowledge is based on morphology. The molecular analysis has been pretty limited. It's mostly based on lineages that have pest species in them. Most of the broader studies have been very limited in terms of the taxa and the DNA regions. So relationships among the higher taxa and composition of most of them are still poorly understood. Monophyly of the subfamily is not certain, and relationships among genera within most tribes are not so clear. Keith already covered the methods pretty well, so unfortunately don't have to do that. And so in this project, we're attempting a broad phylogenomic approach. We have data from transcriptomes, genome sequencing, and also anchored hybrid enrichment. Apologies, we don't yet have a single analysis of the entire data set. A lot of this data is brand new, just got the results in last month. So this presentation will show part of the tree that Keith was showing, as it's got data from all three of the methods, so I'm just going to call that the combined tree. And then the second analysis was more focused on Tephritidae, includes more Tephritidae taxa, but not all the ones that were in the combined analysis. And I'm just going to call that the AHE tree for simplicity. So most of our samples are recent alcohol specimens, but we do have good results from some of the dried specimens we've tried. So that may help us to fill in more of the gaps in the sampling. But we do have pretty good coverage already. We have 2 genera of Tenostylidae. We have 18 genera of Pyrgodidae, including representatives of all the subfamilies and tribes. We have almost 40% of the 500 Tephritidae genera, including representatives of all 7 subfamilies and most of the tribes and subtribes. So this is the tree that Keith was showing. I'll just be referring to the part in the red box there. This is the AHE tree, some of the statistics for it. It's based on 281 loci that are represented in at least 90% of the taxa. So here's the results. In both of the trees, the Tenostylidae, Pyrgodidae, and Tephritidae form a monophyletic group, which is sister to the Platystomatidae. In the combined tree, the Tenostylidae are the sister to all the Pyrgodidae plus the Tephritidae. Toxopyrgoda, which has been placed in the Pyrgodidae, plus Ceratodacus, which has been in the Tephritidae, then form the sister to the rest of the Pyrgodidae plus the remaining Tephritidae. In the AHE tree, however, Toxopyrgoda instead clusters with the Tenostylidae, and that clade is sister to all the other Pyrgodidae and all the Tephritidae. I think this relationship is probably better supported by morphology. Valeri continued to include Toxopyrgoda in the Pyrgodinae, but he also pointed out that there are a number of similarities between it and the Tenostylidae. So I think your option B is probably correct in this case. In both trees, the bulk of the Pyrgodidae are monophyletic, but they're nested well within the Tephritidae. In the combined tree, the main Pyrgodidae clade is clustered with Tachynisca, the Tachynicinae, And those two taxa form a sister group to the Phytalmyeinae. The other Blepharonorinae are not clustered with that clade. They're sister to the remaining 4 subfamilies of Tephritidae. And a bit of surprise, Descoleia, which was in the Phrygodidae, but we don't really know where it went. In the combined tree, it's clustered with part of the Tephritidae. In the AHE tree, the relationships are slightly different. The main Phrygodidae clade is sister group to Ortalotripida of the Tachynicinae plus Ceratodacus. I can't think of any morphological support for that taxon, but maybe it makes sense in terms of biology. Phrygodids and tachynissids are parasitoids. We don't know what Ceratodacus does, but has been collected at lights, so maybe it's nocturnal, maybe it's a parasitoid. And then that clade is then sister to the Phytalmyeinae, and those taxa in turn are sister to the remaining Blepharonorinae, unlike in the combined tree. So the clade, including most Phrygodidae, the Tachynicinae, the Phytalmyeinae, and the Blepharonorinae, are then sister to the other 4 Tephritidae subfamilies. In both trees, the clade of the 4 subfamilies, Tripetinae, Dacinae, Zaceratinae, and Tephritidae, which has been called the higher Tephritidae, is well supported. Subfamilies Dacinae, Tephritidae, and Zaceratinae are each monophyletic, but the Tripetinae is not, which is not a huge surprise. In the AHE tree, this was a big surprise to me at least. The genus Natoma came out as a sister group to everything else. In both trees, Theomaya is the sister to the Zaceratinae, and that clade in turn is sister to the Tephritidae. In both trees, the tribe Adramini of the Tripetinae is sister to the Zaceratinae plus Theomaya plus Tephritidae. And then in the AHE tree, the remaining Tripetinae are then sister to the Dacinae. In the combined tree, the clade including Carpomyina, Tripetinae, and Toxotripinae is sister to the Dacinae, but there's another group formed by Callistomyini, Hexaketini, Cymeratini, and Descoleia that are sister to that. So pretty similar, but slight difference in the trees there. I'm going to quickly try to run through relationships in all the subtaxa. Phrygodids, of course, are scarab, parasitoids. It looks like Toxopyrgoda and Descoleia don't belong there. But otherwise, the genera more or less fit the classification, except that Taxorhini is paraphyletic. Tachynicinae are also thought to be parasitoids. We can't say much about them from this analysis because we only have 1 genus in each of the 2 analyses so far. But I don't see any reason to doubt the monophyly, at least of most of them. In Blepharonorinae, both trees, Stratodacus is not clustered with the other genera. This contradicts a unique morphological synapomorphy within the Tephritoidea. It's seen on the anapisternum, but according to these analyses, that appears to be convergence. Blepharonora and Baryglossa are clustered, as we would strongly expect. Phytalmyeinae include some fruit feeders, but a lot of them are saprophages or specialized saprophages. Within this group, it's no surprise that the well-defined tribes Phytalmyeinae and Phasini are each monophyletic. We're not sure about the Apacrocerini, but the idea of the one sample is not clear. But the largest tribe, Canthonevrini, is definitely not monophyletic. Tripetinae is one of the 3 large, diverse subfamilies. They're mostly fruit feeders, but the leaf miners and stem miners and Tripetinae are also here. This subfamily is not monophyletic. As I mentioned, Natoma, Theomaya, and the Adramini don't appear to belong. most of the tribes do form a single clade. Within it, Carpomyini are not monophyletic, but the Carpomyina and the Paratrelliina are. Ragulatus is not monophyletic as suspected. Trypotina is monophyletic, as are the subtribes Ketostomatina and Trypotina, except for one taxon that looks like a problem there. And those taxa then form the sister group to the Toxotryponini, which is Anastropha, and the sister group to that clade is formed by Paratrelliina, Callistomyina, Hexacutina, and Coelia group. Dacini is another really big subfamily. They're mostly fruit feeders, although the Gastrozonini develop in bamboo or grasses. This is probably the most extensively studied by molecular methods. And our results are consistent with the previous studies. All three tribes are monophyletic, and Gastrozonini and Ceratidini are closely related. Zaceratinae is a very small subfamily. There's only 2 genera. We were fortunately able to include both of them, and they do cluster. And they come out together with this genus Teomaya. Tephritidae is the largest subfamily. Most species develop in Asteraceae, but Tephritoidea and the Pleomeliinae, as well as a few other exceptions, are not in Asteraceae. Within this subfamily, the morphologically well-supported tribes Myopatini and Torelliini are each monophyletic, but the Myopatini and not the Torelliini are sister group to the rest of the subfamily. The No-Eetini is monophyletic except for Encina, which was only kind of tentatively included in the first place. Similarly, the Schistopterini so far are monophyletic except for Perierythrum, which was a genus that Amnon Friedberg always argued didn't go there, so he was right. The core of the Cecidocarini is monophyletic, although Neoragulatus seems not to go there. The Acroteniini is almost monophyletic if you add Pyrgotes and Pyrgotoides, although Acrotenia doesn't seem to belong there. The Xiphocini are mostly monophyletic. Utrutini is definitely not, although most of the genera do go in a clade with a bunch of other non-utrutine genera. The largest tribe, Tephritidae, is not monophyletic. Some genera clearly appear not to belong, but many of them are clustered in 2 large clades. The Dithrocini are not monophyletic. Neither are the Pleomeliinae nor Tephriliinae, but they cluster along with Idaspis, Platancina, and Perierythrum. And all the taxa in this clade, except for Idaspis, are non-Asteraceae feeders, which I think is interesting. So conclusions, Pyrgotidae are nested within Tephritidae. Probably their status will need to be changed. Tachynicinae, which was often in the past considered a separate subfamily, definitely nests within Tephritidae and seem to belong. Our results generally support the existing Pyrgotid and Tephritid classifications. Toxopyrgota seems to be a tenostylid. Toxinorini, Tripetinae, and some Tephritidae tribes are not monophyletic. Various other genera seem to need to be moved around. In the future, we definitely need to combine all of our data, of course. And we hope we can add a lot more taxa to further fill out the tree. Anybody is certainly welcome to join the project. Just want to acknowledge all of our collaborators and also Brian Castle, who's done all the AHE work. And a big thanks to Severin for organizing a great symposium.